So she's got a plant coming out from
Yeah.
one of our reagent bottles. That is clever.
Recently, you all invited urban sketchers. Tell me a little bit about this program that you held.
CCMB is known for connecting with different stakeholders of society. And those of you who know CCMB since its older days would know that this place has had a history and culture of getting filmmakers on campus, artists on campus. People like MF Hussain used to stay here whenever he felt like, for as long as he would want to make his art pieces here. So this is when CCMB was in its first decade. We want people to become partners with science and scientists. And that's what really was at the core of the idea that we are trying out here. We want to create more forums where scientists and non-scientists can mingle with each other. So the idea of bringing in artists now to the campus is actually to utilize their own interests and hobbies and connect it with science. And unlike, let's say, going to Golconda, which has a rich history, this place not only has history, it also has present relevance. They have the possibility of talking to scientists in person, understand what they do, and bring it out in the art that they're making. It captures both the chaos and the order in that chaos very well. So he felt that it captures the essence of their lab very well.
Some of them were already biologists who did their masters or bachelors in biology, and they really wanted to come to CCMB. We wanted them to know what is happening. They are also public. They are our stakeholders. So we wanted them to actually know what kind of research we do, rather than just look at the buildings and be impressed by it. We took them to various facilities like microscopy facility. We took them to the Drosophila facility, the spectroscopy facility, the X-ray crystallography facility, and the greenhouse. And we also showed the lab of one of our scientists, Dr. Raghu.
So we also tried that they get a mix of two things. One, they keep on hearing about DNA, genetics, gene editing. So we tried to show them facilities that really work on those and give them a feel of those to them. So they had an experience of talking to a scientist in person.
You see, this was done exactly standing over here, and this lady has replicated this view. Our campus is filled with art and sculpture everywhere, and some of the artists got inspired by that also. So they have drawn this person, Neha Jain. She has drawn this that Buddha statue over there, and these two statues as well. And they have also drawn these trees.
These are these three.
Yeah, so this one is there. This is one of our the pencil sketches that we got. So if, this is very abstract art that we have in the campus. One of the many abstract arts that we have. But this one is of a lady playing the veena, which has been captured here, and then there's various other abstract art and another person's head's bust. Oh, yeah, so we are at the canteen, the kiosk specifically. This is where we have our evening chai, and this is a place where students and other staff members, they chill during the tea time. So this has been beautifully be captured by this person called Rosie. And you see here there are people standing on these very tables and having chit chat. There is another pen sketch by Srihariji, who's a veteran artist, who has also done in the same place. Except there is this one extra addition. There is a Drosophila here, which is one of the model organisms that we use for various research. And he has juxtaposed this model organism here because that's also one of the research facilities that they have visited. So he wanted to put that there.
What's this?
So this is our greenhouse. This is where we grow our rice plants for rice research. And as you can see, that we keep this under constant temperature and constant humidity. There was one person who was smitten enough to be able to endure this humidity and made this painting from that angle over there. Yeah. So the rice and the lights and everything have been captured very beautifully in this painting.
This facility houses X-ray diffractometers, where you actually put your protein crystals and try to determine the structure of the protein. And protein structures are very important because proteins, they work as workhorses of a cell. Yeah, the urban sketchers saw the protein crystals. They actually went and looked at the models which are actually depicting the protein structural elements. You can see there is some models are present. They also saw these diffractometers using which we actually determine the structures of proteins. Okay, there's a technique called X-ray crystallography using which we determine the protein structures. As I'm blinking, there are so many proteins that are actually in coordination, working in coordination. There are enzymes, your digestive enzymes which digest your food. This. And she started drawing all these sculptures. Yes.
So as an architect, probably she also connected with the structural part of your work.
That's true.
Was there something like a scientist like you who learned something from them and their work?
Yeah, so the thing is that we as a scientist, we often do not, I would say, go out of the bubble that we have. Like whenever I talk to someone, they are most of the time they are either associated with the work that I do, or they are in the fraternity of the molecular biology that we do. But the thing is that when a third person or outsider comes, and so they bring the perspective that they have. So that also is very necessary to understand what we are doing in a better way. So
was there something that you noticed in that piece of art that was done in your lab which you hadn't actually noticed?
Basically I work in a very small portion of that particular workbench. So basically most of the time whenever I work, I focus on what is in front of me. But he drew that piece in such a way that it was a view from this particular angle, the office. So I usually don't you usually don't observe these kind of things. They are there, but you don't observe it unless someone points it out, right? So that was something which the whole kind of view of the lab, it was very beautiful. One of the artist, he was here, and he drew a very, very beautiful sketch of what our workspace looks like, and I was very, very impressed by the way he portrayed the whole lab. It was kind of messy, but that's the beautiful thing about a workspace, right? If it is not messy, then you are not using it enough. So I think it's very great that he drew it like that.
For many of them, it was a novelty, so they were quite excited to see what was happening here. And they came in 3 batches, and we took them around the lab, told them about the kind of work we do. We work on infection biology, primarily understanding how a bacterium called Mycobacterium tuberculosis. This is the bacterium that causes TB in humans. So we told them about the kind of research we do, and it had to be in simple language because many of them are probably not those with a science background. But they actually understood quite a bit that we taught them about or told them. And we showed them around, showed them some of our work, took them around the lab and told them how we do what we do here. So this was quite exciting for them. And some of them brought their children. They were quite excited to see the lab.
Best use of it.
One person had made the double helix.
Can you explain
This is an artist's impression of the double helix.
And this is a proper pencil sketch. That's why.
They were quite fascinated with the things that we actually showed them DNA we showed them a gel. Samdata, unko maine ek gel dikhaya. So they said, is DNA orange or is it pink? No, it isn't. So this is it's new for them, and I'd love to teach them all of this stuff because it's something that they must know. I told them about antibiotics and how they shouldn't take antibiotics for viral infections.
I was thrilled to see when these sketchers and painters were doing their thing, how excited our own people, our own students were to just watch how a creation is made. And I think that's just joyous.
And this guy, the first one that you showed me, the one who was standing there, he made the entire outline with a fountain pen. So he had a fountain pen with a curved nib. I said, did you curve it yourself? He said, no, this is an artist's fountain pen. So he made the whole outline with the thing, including the bottles and gloves and everything. And then he started to fill them. Oh, very goosebumps. Such tiny things he was drawn with bottles and reagents and kya nahi banaya. And he must have made it in like an hour and a half.